Hello, everyone. I'd like to begin by welcoming everyone to today's MBF Bioscience webinar, introducing SLICE, bringing high-performance 3D light sheet imaging to every lab. My name is Matt Fay, and I'm the product manager for the SLICE microscope system here at MBF Bioscience. I'm pleased to be presenting alongside Dr. Raju Tomer today. Dr. Tomer is an associate professor and researcher at Columbia University. The innovations and discoveries from his lab were instrumental in making the SLICE microscope possible, and we're excited to share that story with you today. He brings leading expertise in light sheet microscopy, having developed several groundbreaking technologies, including the Light Sheet Theta Microscope. During his postdoctoral work at the Carl Deisseroth Lab at Stanford, he contributed to the development of clarity tissue clearing technique and created the COLM, the Clarity Optimized Light Sheet Microscope. We're honored to have Dr. Tomer join us for this webinar, and I want to thank him for being here today. Just a couple logistical things to get out of the way first. You can feel free to ask questions during this webinar using the Q&A button near the bottom of the Zoom window. This will open the question and answer panel. You simply type your question and press enter, and it'll be submitted to us. Dr. Tomer will begin today's presentation by discussing the need for more accessible light sheet microscopy within the research community and the types of scientific questions this technology can help address. Dr. Tomer will then share the breakthrough innovation developed at his lab that significantly reduced the complexity and cost traditionally associated with light sheet microscopy. Following that, I'll introduce the SLICE light sheet microscope, highlighting key features and specifications, and provide an overview of the $75,000 package being offered by MBF Bioscience. Then we'll walk through a demonstration of how to set up a tissue imaging session using the SLICE and showcase a final image, giving you a glimpse of the results you could be achieving in your own lab. Finally, Dr. Tomer and I will be available for a Q&A session at the end of this presentation. I'd like to now turn it over to Dr. Tomer to begin his portion of the presentation. Dr. Tomer, thank you.
Thank you so much, Matt. Also, thank you, MBF, for organizing this webinar and also giving me the opportunity to present our work today and share widely. Let me jump right in and start with a sort of a big picture overview of our motivation in our lab, or the research in our lab. As we all know, in the last years, there has been tremendous progress in application of AI, machine learning methods, and tools in biology. And this is in part driven by, of course, the development of scalable architectures like transformers or vision transformers, and also availability of GPUs for training of deep neural networks. But in large part, also a result of availability of large amounts of data in sort of a structured, usable, machine-readable format, right? Which really allow us to train these deep neural networks, right? So, so for example, for protein structure prediction, right, the tools like AlphaGo, AlphaFold, and lately the ESM models, right? They are all developed by building upon the tremendous amounts of data on protein structure that has been generated over several decades, right? Similarly, now we're talking about developing virtual cell generative models. That's also driven by availability of extensive amounts of omics data, genomics, proteomics, epigenomics, single cell RNA sequencing from different cell types in different tissues in different cell states, right? So I think it's fairly clear now that going forward, having large quantity of data in sort of a structured machine readable format can be the driver for sort of big innovations in the life sciences field. So the beyond single cell models was the next frontier would be try to develop organ scale or even organism scale foundational models, right? That could be used for in silico experimentation. And of course, to reach that goal, we'll need tremendous amounts of three-dimensional image data from diverse types of tissues in diverse states diseased or normal or other conditions, right? So I mean, that's what really motivates us. So we're going to help that process by developing tools that are scalable, that can be used widely, and sort of software ecosystems that can be used to sort of standardize these data so that they can all be pooled in to eventually train these deep neural networks. So for the 3D imaging, it has become clear over the last years that the light sheet microscopy is kind of the method of choice. Simply because of this unique geometry, it was discovered more than a century ago, right? That you use a thin sheet of light to illuminate a thin plane in the sample, and the signal from that is detected with an orthogonally arranged, typically a wide field arm, right? So the end result is you end up doing very gentle, high-speed imaging, right? Which is what we need for imaging large cleared samples, but also like live samples. If you want to do gentle imaging over a long period of time. So already for last couple of decades, light sheet microscopy is driving great advances in many different domains. So I just want to share one example from our own work. So we use light sheet microscopy for sort of a whole brain, unbiased investigation of how drugs impact brain-wide neural networks. Right? So in this case, focusing on antidepressant ketamine and how exposure to that over time impact the dopamine system. So in the rendering on the left, what you're seeing is sort of changes in the neuronal cell body density, and the rendering on the right is showing changes in the brain-wide projection of dopamine neurons. Orange is the increase, and cyan is the decrease. Right? So it's really clear that the power we get from such unbiased whole brain studies that you can discover things that would normally, with targeted studies, we cannot. Right? So here we can see the impact of ketamine is really divergent, right? In some areas, there's a positive impact. In other areas, it's a negative impact, right? So it again comes to the point when we take a drug for some brain disorder, right? It might be targeting some areas for positive, but there could be serious unintentional impacts on other brain areas as well, right? Because the drug is kind of delivered uniformly throughout the brain. But anyway, so light sheet microscopy is allowing such detailed investigation at systems level. To do that study, we had to really image lots of brains. So we in this particular study, we imaged 95 whole mouse brains, about 100 terabytes of raw data that needed to be analyzed, right? And the images needed to be at a high enough resolution that we can kind of look at the projection pattern, brain-wide projection pattern, right? Unfortunately, such high performance light sheet microscopy capabilities and data analysis capabilities remain inaccessible to most researchers. Right? It's restricted to either the optics expert labs or well-resourced labs that can afford, or sort of advanced imaging core facility. So there are several challenges that need to be addressed to make it more accessible to masses, right? At a broad scale. The systems in general still remain prohibitively expensive. Commercial systems typically can be several $100,000 depending on the company and specs that you get. Even the high quality open source projects like Mesospin can have material cost in hundreds of thousands of dollars, right? And then of course you have to build it or get help to build it, right? So it's not really something that easily adoptable by, let's say, non-experts, right? And the systems in general still remain non-portable complex, so it's getting better and better. I mean, light sheet microscopy is still not easy to use by non-experts like clinicians. Right? It needs some sort of understanding, as opposed to confocal or slide scanners, right, which have become more or less push-button system with full automation. Light sheet microscopy there's a barrier on the usage, right? And then there's a severe lack of standardization in tissue processing protocols and data formats and data specs, right, meta information and whatnot, which needs to be standardized to eventually allow us to pull in data sets generated at different parts of the world, right? So that we can have sort of a more diverse representation in the training data sets when it comes to, let's say, training AI models or for local studies. All right, so in our recent work, we tried to address some of these challenges by developing a low-cost, scalable light sheet microscopy platform that we call Projected Light Sheet Microscopy, while still maintaining the high imaging performance than the conventional high-end system, or even going better than them, right? So the basic idea here was really that we asked the question, can we use off-the-shelf consumer-grade components, which typically are optimized for cost and performance, can we use them to build a system which is useful in a real-world scenario, right? Not just as a toy or as an educational instrument, but something that allows real-world experiments with high imaging performance that is needed for various experimentation. And answer to that is yes. So we found that we could use this cheap $300 pocket laser projectors to generate high quality, powerful, with enough power, thin sheets of light that we can use for 3D imaging in a light sheet microscopy format, right? In this work, we also used consumer grade CMOS chip that are much less expensive and still provide good enough performance, right? So together with other optical innovations, optimizations there, we ended up with this highly compact device with minimal mechanical, optical, electrical, and also computational footprint that is also magnitude cheaper, but still provide imaging performance at par with the state of the art, or even going better, as we'll discuss today. So in the next few slides, I'm going to present some technical characterization of the system and also some data points to kind of make the point that we indeed get high imaging performance, and it's useful for many experimental scenarios. So here's just the starting example of typical use case of imaging whole mouse brain. So it's this brain is stained for vasculature, and that was imaged by the pLSM system. Okay, so looking a little bit into the technical aspects of the pLSM, let's start with the sort of the resolution aspect. So we characterize the PSF, or the resolution of pLSM system, by imaging sort of sub-resolution fluorescence beads, right? Using two different objectives. So here, this is air detection objectives, 10x 0.28 NA, with long working distance, 34 millimeter working distance. So with this objective, imaging in a sort of imaging an immersed sample, we achieve lateral resolution about 1.8 micron or so, right? The axial resolution about 5 to 6 micron, with pretty good field of view. We also found that this platform, in principle, is also compatible with the, let's say, optimized immersion detection objectives. So in this case, 16x 0.4 NA, I think with 12 millimeter working distance. So with this, we can of course get compared to this objective in this paper, resolution about 0.85 micron, 0.9 micron lateral resolution, and the axial resolution is of course similar. So I'll come back to this point again. Let me move to another aspects of it. So one common experimental goal is to do, of course, multi-channel imaging. So we demonstrated that we can use the red, green, blue lights of these pocket laser projectors to do multicolor imaging of common fluorophore combinations that are used in biology. So here is one example demonstrating that, of three common fluorophores combined, EGFP, Alexa 555, and Alexa 647, that we can image in multicolor, right? And I mean, this is one example, but the fluorophores tend to have pretty wide excitation and emission spectrum. So with using the right emission and excitation filter, we can cover almost any fluorophore, and I mean, even can go beyond 3 if we want to. Right? That's a possibility. Of course, we haven't done that. Another kind of problem in imaging of large cleared samples like mouse brain is that the alignment between the light sheet and the detection focal plane, it can change in a sample position dependent manner, right? So for example, you align your light sheet with the focal plane at a particular, let's say, particular depth, Z, and you go 1 millimeter deeper into the tissue, and then you see it's completely defocused, right? So that's a challenge for achieving high quality imaging of large samples. So in this work, we found that by choosing few anchor points in three-dimensional space and finding the optimal alignment parameters there, and then using a simple linear interpolation, we can develop a lookup table that can be used to achieve high quality in focus imaging throughout large samples, right? And this approach also has very nice additional advantage that it makes the system sort of multi-immersion compatible. So now you can use it to image tissues from various different tissue clearing methods. So in next few slides, I'm going to show some data points along those features. So starting with the multi-immersion compatibility. So here's a example of imaging whole mouse brain processed with iDisco clearing method. So this is a iDisco processed brain stained for tyrosine hydroxylase, which is a marker for a dopamine neuron. And on the right side, you see that this data we can analyze with the standard segmentation tools and also register it well in Brain Atlas to get this brain-wide distribution of the dopamine system in this case. So iDisco clearing typically has effective index of 1.52 to 1.55 depending on variant that you use. Here's an example of showing of imaging clarity cleared sample, which typically has refractive index in 1.45 range, so different refractive index, and without any hardware change because of the nature of the adaptive parameter correction. We can image this sample also at high in focus quality throughout. Right? All right, so we also demonstrated the system is compatible with really large tissues. So in this example, showing the imaging of chunks of human brain. So in this first example, this chunk of human brain is stained with antibodies against podocalcin and ACTA2 to label the vasculature. And here on the right, you can see sub-volumes, small volumes at different depth in the sample. So this is more or less on the top, this is middle, and this is at the bottom, imaged from one side, right? And as you can see, we achieve great quality, great contrast, great resolution, and the data is amenable to analysis by, again, standard approaches. So in this case, reconstruction of the vasculature networks, and you can quantify typical aspects of it, like the diameter of vessels, as well as the branching patterns and the density and so on. This is my favorite example of imaging human brain, postmortem human brain from Alzheimer's. So here we are mapping the distribution of amyloid precursor protein in Alzheimer on the top, and then there's sort of a neurotypical cortical sample. And as you would expect, there's a lot of aggregation of the APP in the Alzheimer's. It seems to be a pretty severe case in this case. So we can quantify these kind of details in very large samples in high quality. All right, final example I want to show is about compatibility with high throughput imaging. System is compatible. So here we demonstrate imaging of a bunch of brain organoids cleared and stained. So in this case, they're mounted on a sort of agarose-based embedding in a sort of a vertical line, and then they all can be they all are imaged in one single experiment, one single imaging experiment with good enough quality that you could do segmentation. And so this is a nuclear label SOX2, right? You can segment it and you can generate this point cloud, quantitative point cloud about the expression levels of that particular marker. This is here just showing you zoom in. This is the raw data, and this is the point cloud reconstruction. Then you can, of course, do all sorts of quantification in terms of number of cells, cell density, the signal level distribution across cells, across depth, and so on. This is sort of another similar example of imaging a bunch of human vasculature organoids. And so they're stained with two antibodies. So it's a two-color imaging, allowing us to kind of look at different aspects of the vasculature in terms of their connections, junction strength. All right, so that's that was the work we did last year. That was published last year, and we demonstrated that it's possible to use off-the-shelf consumer grade components to achieve high performance imaging at a fraction of cost of the state-of-the-art or the conventional imaging system that are available. Since then, we have further advanced the technology, and those are also implemented in SLICE, as Matt will show. So I want to discuss one specific advancement in that context that has to do with the resolution. So as I discussed before, there's a great advantage of using air objective for imaging of large clear tissue. First of all, they're cheap, right? They're orders of magnitude cheaper. And they typically have very long working distance. So this objective has 34 millimeter working distance. And they're generally very well color corrected as well, right? So like objectives coming from machine vision field. But the one problem is, of course, we use air objective to image a sample in immersion media. You end up with aberration, typically spherical aberration, which, of course, reduces the resolution and also the signal collection, right? The amount of signal that you collect. So we really asked the question that can we kind of use air objectives and get the performance which is as good as or better than these highly optimized immersion objectives, while keeping the good features of air objective of long working distance, right? So that we can do really deep tissue imaging. And answer to that is yes, we can. So the problem with the air objective, as I mentioned, is you end up with spherical aberration if you're imaging a sample in immersion media. So we basically designed a strategy with sort of building in correction elements in the sample chamber itself in a modular way. So we have a manuscript in preparation to describe this that allow us to basically fix all these aberrations. So the bottom line is we end up with 0.75 to 0.8 micron lateral resolution over large field of view while still keeping majority of the working distance of the air objective. So coming back to this again, the advance really is that with the orders of magnitude cheaper objective with longer working distance, we can essentially get performance better than more expensive immersion objectives with comparatively smaller working distance, right? So we're achieving about 0.75, 0.8 micron, which is already much better than what we can get with these objectives. All right, so that's implemented in SLICE as well. So I just want to end with showing a sort of a latest data example used by imaging that improves systems. It's work by Pauline. She's working on to combine SLICE or pLSM with expansion microscopy to basically eventually reconstruct neural circuits at the whole brain level. So this is a expanded mouse brain sample, Taiwan EGFP, so it's expressing EGFP. This is just recently acquired data, so we're still working on kind of doing the proper stitching and so on. But just to show a little bit of the quality of the data that we get, here is a zoom in. So this is a neuron, and all you see is dendrites, right? And then you can see dendritic spines level details. This will become actually much clearer in this sort of a small slice by slice stack movie. Again, it's imaged by 10x 0.28 air objective, and we essentially can capture dendritic spines level detail, and in a comprehensive way, right? Like covering the entire spine, saying not only like here and there, right? Okay, so with that, I'll finish. Hopefully, I made the case that it's possible to achieve high performance imaging, light sheet imaging with real world applications with consumer grade components, hence reducing the cost by orders of magnitude, and also kind of further improving on its footprint, right? Really decreasing the mechanical optical footprint to make it more portable, more user friendly. So I'm very grateful to have collaborated with MBF to kind of take pLSM to the next step to build a much more advanced, much more robust and user-friendly version, SLICE, which Matt will talk about. So with that, I'll end, and thank you so much, everyone, for your attention, and I'll hand over to Matt. Thank you.
Thank you, Dr. Tomer, for that discussion. When the Tomer Lab approached MBF Bioscience with their innovative light sheet technology, we immediately recognized an opportunity to democratize discovery, to bring high-performance light sheet microscopy to every lab. But we also knew that affordability alone wasn't enough. The system needed to deliver the same or better level of performance and image quality researchers expect from a core facility instrument. We understood that enabling all researchers to image intact organs and whole organ samples without the need for sectioning would unlock a new range of scientific possibilities. And to make those experiments a reality, we knew the system had to be flexible, giving the researchers the freedom to choose the tissue clearing method that best suited their samples and scientific goals. The development of the SLICE continued at MBF Bioscience with the significant help of Dr. Tomer. We made this system compact to minimize the space needed in the lab. We re-engineered the light sources to have 2 times the laser diode intensity of the original design and drastically increased the efficiency of light sheet delivery to the sample. This meant faster and more sensitive scanning of samples. We made sample positioning faster and more accurate with a closed-loop linear encoded XYZ piezo stage. This made accurate positioning of multi-field of view acquisitions possible. A motorized filter wheel gives us automated multi-channel imaging capabilities, which is important for visualizing distinct structures in light sheet microscopy. Increased detection sensitivity with an 86% peak quantum efficiency backside illuminated camera and a maximized usable field of view with a 26 megapixel sensor. And with the help of Dr. Tomer, we introduced the corrective optics that he discussed earlier to improve the system's resolution. The design changes we just discussed enabled SLICE to have lateral resolutions of 0.75 micron, giving us subcellular resolution with a 10x detection objective. Our axial resolutions of 5 to 8 micron, depending on the illumination configuration you choose, give you sufficient resolution to quantify morphology or detect cells within samples. Our field of view of 1.2 by 0.75 millimeters with a 10x detection and a scan range of 20 millimeters by 20 millimeters by 18 millimeters is sufficient for acquiring mouse or rat brains and large samples. Our imaging speeds of up to 17 frames per second, depending on sample and exposure time, and excitation wavelengths of 450 nanometer, 520 nanometer, and 640 nanometer align with common fluorophores that researchers choose to use with clear tissue imaging. Our system dimensions are 48 by 37 by 31 centimeters. This makes the SLICE about the size of a typical microwave oven. The BrightSLICE software is included with every SLICE microscope system. It features everything you need to control the microscope with easy-to-use workflows that allow the system to be operated by an expert or a novice user. 3D stitching and post-processing filters to make your multi-tile acquisition into one final seamless image. 2D and 3D visualization for inspecting and generating publication-ready images or movies. Efficient data storage and management pipelines to keep your image data safe and accessible. MBF Bioscience's JP2000 image format allows for compression of images without introducing artifacts. This can make the most of your data storage and take a data set that is tens of terabytes to only hundreds of gigabytes or less. The $75,000 SLICE package comes with everything included for your lab to start acquiring light sheet data. The SLICE Light Sheet Microscope system with BrightSLICE software to acquire, process, and visualize data, a high-performance workstation with an AMD 16-core processor, 128 gigabytes of RAM, and a 20 gigabyte graphics card give you everything you need to visualize and analyze your data. A 4K high-resolution display, and right now, when you buy SLICE, you get a free 6-month trial of the MBF Bioscience's Light Sheet suite of analysis products. When you order a SLICE, you'll receive a free 6-month trial of MBF Bioscience's Light Sheet suite. With this suite, you can register brain images to the Allen Brain Atlas and detect cells by regions using NeuroInfo, reconstruct complex neuronal morphology with NeuroLucida 360, or analyze and reconstruct vasculature using VesaLucida 360. Also not shown here, you can perform unbiased stereology to accurately estimate population, length, or volume with Stereo Investigator. All of these tools are available for you to explore during the trial when you order a SLICE today. I want to briefly touch on tissue clearing, a transformative technology that expands the capabilities of light sheet microscopy. Biological tissues are naturally opaque, which leads to light scattering when imaging large volumes. A major contributor to this opacity is mismatch in RI, or refractive index, between different tissue components. As light passes between materials with different RIs, it bends or refracts, distorting the image. Tissue clearing addresses this by homogenizing the refractive index throughout the sample, allowing light to pass through with minimal light scattering. On the right, you can see an example highlighting the impact of RI matching in a cleared tissue sample. There are many clearing techniques available, such as Clarity, iDisco, and Cubic R, each with its own target RI. Many light sheet microscopes are optimized for a single RI, limiting their compatibility. But SLICE is different. It's designed to handle all clearing techniques, making it one of the most versatile systems available. SLICE is able to image these various sample preparations independent of their RI by adapting the light sheet's position through software control. Through the adaptive RI compensation feature, we set up points of focus in the sample, and the software will interpolate the light sheet's position while scanning live and during acquisition. On the screen here, we have an iDisco cleared tissue sample with an RI of approximately 1.56. Here we're showing the same field of view with and without RI compensation while moving deeper and deeper into the tissue sample. Very quickly, you can see the image on the left loses resolution. However, with RI compensation, we're able to maintain crisp focus and resolution while imaging the full volume. On the same day, we imaged a clarity sample with an RI of approximately 1.46. We didn't change any hardware, only the sample that is on the stage. Without RI compensation is shown on the left again, and with adaptive RI compensation is shown on the right. Again, you can see as we move through the volume, we quickly lose focus, but with SLICE's RI compensation mapping, we maintain focus throughout the entire volume we're imaging. For the majority of homogeneously cleared tissue samples, 2 focus points throughout Z is all you need to correct for this change in refractive index. However, we can still image samples that have heterogeneity in terms of RI by using our XYZ RI correction. We'll see an example of this in the imaging demonstration next. Let's now look at the BrightSLICE software. We'll begin with a quick tour of the user interface. The large central area of the screen is dedicated to live image viewing and 2D image viewing. To the right, you'll find all the controls for our light sheet excitation and our emission filter wheel, a software joystick to control our stage and move our sample through the field of view, our RI compensation tool where we'll add points of focus throughout the sample. A camera histogram displays our lookup table and dynamic range, and camera settings to adjust our exposure. The multi-channel control allows you to save all of your light sheet and camera settings to a preset, making switching channels just the click of a button. Now let's walk through how to set up an acquisition. While setting up acquisitions, I like to keep my illumination intensity low and camera exposure high. This prevents photo bleaching as we're navigating around the sample and setting up our acquisition. It may cause a high background appearance while setting up, but that preserves the fluorescence of our sample for our acquisition. You'll notice that the sample appears out of focus in this current field of view. To bring it into focus, we isolate one light sheet and adjust the offset for that corresponding light sheet. We'll then repeat the process on the other light sheet. Once we're creating the same image with both light sheets, we know that they're coplanar, and we can add this point of focus to our RI compensation tool. Once we have multiple points added to our map, we can start interpolating, and SLICE will adapt the offset while imaging live and during the acquisition. Once I have my adaptive RI compensation, I like to navigate around the sample, making sure everything is in focus. This is a great opportunity to place the markers that we have in the BrightSLICE software at the XY extents of our sample, so we can use them later when setting up the acquisition in the workflow. Here I've reached the left edge of the sample, and all I need to do is select a marker from the left toolbar and place it within the imaging environment. I'll then repeat this process for the upper extent of our sample, the right extent of our sample, and the lower extent of our sample in XY. It's important in this process to ensure you're moving through the sample in the Z imaging axis as well, to make sure the tissue does not extend beyond the field of view you're looking at in an imaging plane above and below. You'll notice that as you place these markers in the imaging environment, they will display in our macro view tool. This tool shows you all of the markers in your imaging environment and makes it easy to return to them later. Now that our sample is delineated and looks in focus with our adaptive RI compensation, we'll jump into the tissue scanning workflow. We'll choose to set up our XY area by placing markers. We can use the Go-To feature of the macro view to quickly and easily return to the markers we placed at the XY extents of our sample earlier. Once we place these markers, you'll notice that our XY scan area will display in the macro view. Moving on to the next step, we need to set the depth extents of our volume. By navigating with the stage's Z-axis, we can move to the top and the bottom of the sample and press Set for each in the workflow. In the next step, we have the option to choose what type of RI compensation we're going to use. Since we've already set up a Z interpolation compensation, and our sample is looking great, we'll select that and move on. In this next step, we have an opportunity to run a quick sample acquisition. This is a chance to run a quick scan using the settings you have set up to make sure everything is imaging well before acquiring the entire sample. In the final step, we need to choose a save location for our data, name the file, and start scanning. Here is a video rendering of the intact mouse brain we just set up the acquisition on. This mouse brain was cleared using the cubic R clearing technique and is from a FOSS trap mouse, expressing TD tomato fluorophore. We can see the TD tomato being expressed in the somas of neurons that were active before the mouse was sacrificed. Additionally, we do see some autofluorescing vasculature in this sample as well. This particular researcher is then using MBF Bioscience's neuroinfo analysis software to register this brain to the Allen Brain Atlas and perform cell detection by brain region to quantify activated cell populations. This image was acquired with a 10x over 0.28 NA imaging objective and an 8 micron axial resolution. This video may appear jumpy over the Zoom screen share, but it's available on our website if you'd like a closer look. In conclusion, light sheet microscopy is an incredibly powerful imaging technology, but for many in the research community, it's been out of reach due to its complexity and high cost. SLICE democratizes discovery by bringing high-performance light sheet microscopy to every lab. It delivers the imaging power you'd expect from a core facility in a system that's compact enough for the benchtop, versatile enough for a wide range of experiments, and most importantly, it's affordable. SLICE is a complete end-to-end imaging solution. You can begin acquiring light sheet data in your lab today, and with your purchase, you'll receive a free 6-month trial of MBF Bioscience's image analysis software, all for $75,000. I'd like to now open up the floor to any questions for Dr. Tomer and myself. Thank you, everyone, for joining. We had a few question submissions before the webinar. One question was about how many fields of view it typically takes to acquire a mouse brain. We do have the 10x detection objective. With that objective, it really depends on the size of the sample and the clearing technique that's being used. With the clearing, there can be slight expansion or shrinkage to the sample. But somewhere between 8 and 9 imaging tiles over the course of maybe 1,000 to 1,500 Z planes through the volume for a 10x objective. For a 5x, it's typically around 5 to 6 tiles with the same Z sampling. I think there may have been another question about whether this system typically needs a optical table or a vibration isolation table. We have not found the need for those in most environments. We can set the system up on the benchtop, and the built-in vibration isolation seems to be sufficient in most cases. If there is a need for vibration isolation, say you have some machinery running in your lab facility, we can provide that as well. I see a question in the chat about training available. We have a great user guide, and yes, training comes with the purchase of the slice. We have a session that helps you get your system set up in your lab, and then we'll go through training you and help you with your first acquisitions, and then support is always available. We have a great support team and a subscription that you can subscribe to, and you can reach out to support anytime. See another question here about how to mount whole mouse brain samples. So on the slice, we use cuvette-based mounting systems. So we're just using standard off-the-shelf cuvettes that we've customized attachments and components to couple those cuvettes to our stage. We have different sizes of the cuvettes that are going to be available. We have our standard size that we offer is just a 1 centimeter cuvette, 1 centimeter square by about 42 millimeters. Typically fits most mouse brains unless there's a little bit of expansion going on. We have a 10 by 5 millimeter cuvette, as well as a 10 by 20 and a 20 by 20 cuvette available. We also make what we're calling the brain brackets. These are, just little mounts that you can mount your sample onto, and then put that mount with the sample oriented correctly into your cuvette for mounting. Typically, a little bit of adhesive helps. We've found silicone is a great choice. Crazy glue has also been used, as well as dental cement. See another question here about the image formats. So the raw data, we actually use a raw data format. Our final image format is a JP2000 format. That's a great format to use. It works awesome with all of our MBF Bioscience analysis products, and we can also perform that compression on it, making sure that we are making the most of your data storage and costly drives that it can take to store all of this light sheet data. We do have the capabilities in our software to save as an OME TIFF, and also have a free headless conversion tool that will do that conversion for you. See another question here about how fast this microscope can acquire or scan a mouse brain in 2 channels. Typically at 10x, where it takes about 4 to 6 hours to acquire each channel. So you're looking at something around the range of 8 to 10 hours, 12 hours, depending on the size of the sample. If it's an iDisco cleared sample, maybe it has some shrinkage. It would likely be shorter than that, but I've seen some clearing techniques expand a mouse brain to look more along the scale of a rat brain. So um, typically for 2 channels, you're looking at about 8 TB of raw data. After we've compiled that in our JP2000 format, we can get that down to hundreds of gigabytes without losing any resolution or compromising any analysis detection and quality. I see another question about the size of the cuvettes. So the cuvettes we're offering are a 10 by 5 millimeter, a 10 by 10 millimeter, up to a 20 by 20 millimeter cuvette, which pretty much covers the full scanning range of this slice microscope at this time. See another question here about taking 3D images of other tissues like the spleen. Yes, you can definitely use the slice for many different types of tissues. If you're able to clear it, that typically helps if you want to image the full volume. But really any organ up to 20 by 20 by 18 millimeters is the scan range we can achieve on the slice. See another question here about the Z points for the RI compensation. Typically what I'll do when I'm setting up something like a mouse brain on the slice is I'll go towards the top of the sample, maybe not the very top of the sample, and I'll put a focus point there, and then I'll go down towards, the bottom of the sample in Z, and I'll make that in focus as well. I'll add both of those points to my map, and I'll see how my interpolation looks at that point. You can turn on and off the interpolation as needed. So as you're navigating around the tissue, you can tell if things are out of focus, or you can adjust your offset to see if you can make a certain portion of the tissue that you're looking at in your field of view more in focus. If you need more points, you can then add them at that point. But I typically don't like to add any more points than I need, since it takes time to create those focus points. Typically for a mouse brain that's homogeneously cleared, I put two points spaced apart towards the top and towards the bottom of the sample, and that's been sufficient. See another question here about lower power detection objectives. So right now, the lower power objective that we can offer is a 5x magnification. We are in search of other solutions, but those are not readily available. Dr. Thorick, I see your question here. Thank you for joining today. I can comment on the sample fluorescence. In the context of, how much light you want to use, it's typically what I do to determine how much light can I use is I'll slowly increase the amount of light until I reach a point where I'm slowly photobleaching the sample. Every fluorophore is slightly different. Typically dyes like Alexa dyes or other available dyes tend to be more robust and resistant to photobleaching, whereas certain proteins like TD tomato or EGFP, those tend to be more sensitive to light. So it really depends on the fluorophore that you're using in your experiment. I would recommend starting low with the light and just slowly increasing until you find where that barrier is. Then when I'm setting up my sample, I usually like to lower my light even beyond that, just to make sure that if I do end up sitting in one position for a moment or so, that I don't end up bleaching that portion of the sample. You can always increase your exposure while you're setting up your acquisition. And that will give you a similar looking image quality and give you enough data to create those compensation maps and set up your acquisition.
If I can add briefly to that as a sort of also practical advice, I mean, we typically go by gray levels, right? So you increase the power, and then you have sort of your minimum gray level and the high gray level, and typically a gray level range of 1,000, 2,000, right? Going from minimum to maximum is sufficient for most analysis, right? You get enough dynamic range. So of course you want to use as low power as you can, and how much low you can use is kind of depend really. One way to decide that is by kind of looking at the gray levels you're getting, the dynamic range you're getting from low to high. And generally, for live samples, I would keep 200, 300 gray levels if you want to minimize. But for clear samples, like 1,000, 2,000 from minimum to max, that that that's sufficient for, let's say, analysis part, right? You have enough dynamic range in the system. But yeah, it's a sample dependent process there.
Thank you, Dr. Tomer. I see another question here about, can the system accommodate different objectives and can they be interchanged by the users? Yes, it can. We can do, customization in terms of the illumination objectives. We can increase the magnification that's offered with the base package, which gives you actually a thinner light sheet. So that can be changed out as well as the detection objectives. So for detection, we offer a 10x objective as well as a 5x objective. Those can be changed by the users. We have a very helpful alignment workflow that is about to be released in the coming weeks. So there will actually be a workflow that will take you through the process of realigning after you've changed those objectives. It's a very quick and easy process to follow along with the workflow. It maybe takes maximally a half an hour once you get the hang of it. I've changed objectives in 10 minutes as well. So it's not a very complicated process. There's no realigning, very difficult to align optics. It's a really, really easy process to do, and that will be available for users to change out and put objectives in that fit their imaging needs. See another question here about can other tissues be or cleared too? Yes, this is not just mouse brains that can be cleared. Many different tissues, many different organs. I've seen customers clearing bone, even decalcifying bone and clearing that as well. We've imaged hearts, we've imaged lungs, kidneys. The possibilities are really endless, and it's just about finding the right clearing technique for your experiment. See another question here about how long to scan a whole mouse brain. It really depends on the mouse brain and the clearing technique that you're using and what detection magnification that you're using. Acquisitions could be as quick as an hour or two, but the system is designed to run for days on end. So no matter how long it does take, the system is reliable and you can scan as long as you need to acquire your data. So it could be anywhere from a few hours to something like 8 hours, depending on the sample and the fluorophores that you're using. I see a question here about on-site demos. We are hoping to ramp up to offering more and more on-site demos as systems become more and more available. The demand's somewhat high right now in terms of on-site demos. So the easiest thing that we can offer is we do offer evaluation tissue imaging. If you'd like to send us a sample, we're happy to image it and provide you images. And then we can also do remote demonstrations, which may happen more quickly than an on-site demo. But if you're really interested in an on-site demo, that's something we can reach out to you and set up. See another question here about, can the light sources be replaced by an end user in the field? I would not recommend doing this. It would be a difficult procedure to achieve, and the light sources that are included in the slice are really optimized to make the slice this high performance imaging engine that it is. So I would not recommend doing this in the field. I see another question here about support on the clearing process. It really depends on the clearing technique that you're using. We have a very knowledgeable support team that's happy to, assist you with your imaging experiments within their abilities. We do not perform clearing ourselves. So we're happy to assist you as much as we can in that process. But it is not something that we make products for or provide in-depth technical support on. See another question here about light sheet acquisition systems and the images they generate not integrating well with other analysis tools. The MBF Bioscience package is a full end-to-end solution. So the slice images that you generate are, seamless integration into all of the MBF Bioscience software. If there's another analysis product or software that you really would like to use, we do offer the ability to save whatever image format you need to get to. Typically, we convert to an OME TIFF, which is a more generic image format, and that will allow people to um, branch the gap into the analysis software that they need for their experiment. Yes, the software does include 3D rendering and video generation. It's really optimized for visualizing, teravoxel sized data sets. So you can open up your entire mouse rat brain, and that loads in a matter of seconds. It's very impressive, and then you can generate videos and other things that you need from your publication. That video generation and visualization is available in all of MBF Bioscience's analysis products. So depending on if you're just using the BrightSLICE software, you get all the visualization and the movie generation. If you have the analysis products as well, you get the same visualization and movie generation as well as the analysis that's included with that product. Like to reiterate all my thanks for you taking the time to join us today. If we did not get a chance to answer your questions, you can reach out to us at info@mbfbioscience.com. Please don't forget to fill out the short survey at the conclusion of this webinar. Your feedback is an important component of our continuous improvement process. For general questions, discussions, FAQs, we would like to remind you that we have the forums on our website where users can communicate with each other and other MBF experts. This is forums.mbfbioscience.com. We welcome all of your input and look forward to hearing from you. We also invite you to join our weekly MBF Bioscience open office hours. It's a great opportunity to connect directly with our scientific and technical experts and ask questions, as well as get tailored advice on your specific research. That concludes our webinar today. Thank you everyone for joining and taking time out of your day.
Thank you.